Hi all, thank you for attending the School of Health and Biomedical Sciences 3 minutes thesis competition for 2025. Before we start, I would like to welcome you by acknowledging the people of the Woiwurrung and Boiwurrung language groups of the Eastern Kulin nations, on whose unceded lands we conduct our business. We all pay our respect to their ancestors and elders, past, present, and emerging. RMIT University acknowledges the traditional owners and custodians of the land and water, of the lands and waters across Australia, where we conduct our business. I'm Associate Professor Vassilios Stavropoulos. I serve as the Associate Dean for HDRs for our school. And I would like to remind to all of us why we are here today. It's an opportunity to celebrate HDR research. It's a celebration more than a competition. The idea was initially conceived by the University of Queensland, and it aimed to foster communication and research presentation skills. So it's more of a celebration, I reiterate, than a competition, and has some very specific rules, which is presenters have only one static slide. They present only via words, they talk. There are no props, no electronic media, and they have exactly 3 minutes to present. Therefore, try to be silent, and if you can, mute your mobiles, because it's going to be extremely helpful for everyone and for the process. Over 900 institutions across more than 85 countries are holding a 3 minutes thesis competition event. And it's a globally recognized competition, which our school had the great privilege to win at the university level last year through Michaela, who is going to be talking to us at the end of this process. I think I covered the competition rules. Now, judging criteria. We have 3 esteemed judges sitting at the back. They are very friendly and very impartial, I would dare to say, and they bring different skills to the table. Judging criteria: comprehension and content. Whether the presentation provided clear motivation, background, and significance to the research question. Whether the presentation clearly described the research strategy, design, and the results or findings of the research. And whether the presentation clearly described the conclusions, outcomes, and the impact of the research. Engagement and communication. The oration was delivered clearly, not like the way I talk, and the language was appropriate. No, I don't mean that like that. And the language was appropriate for a non-specialist audience. The PowerPoint slide was well defined and enhanced the presentation. The presenter conveyed enthusiasm for their research and captured and maintained the audience's attention. Now, our judging panel, Mrs. Ezra Beslen, our Senior Services Officer, STEM Planning and Operations. She's the one who approves all the HDR funds requests and helps you to continue pursuing your studies during your research journey. She's always helpful and kind to us, very generous with her time. Dr. Jack Thean, Vice Chancellor's Senior Research Fellow and Deputy Lead of the Healthy Lifespan and Chronic Diseases Group. Thank you, Jack, for allocating your time to us today. And Associate Professor Oren Tyros, Clinical Biomechanics and Rehabilitation. Thank you, Oren, with extensive experience across different universities, several PhD completions, and goes on. Now, prizes. RMIT hits, winner, $500, runner-up, $150, People's Choice, $150. For the ones who are going to reach RMIT final, the winner, $3,500 research grant, represents the college at the RMIT 3 minutes final. Runner-up, $1,000 research grant, and People's Choice, $500 research grant. Now, heat presentations to begin. Our first candidate, our first presenter, Musab Omar, He's going to present us his project evaluation of a novel BCR, ABL1 tyrosine kinase inhibitor asciminib. I'm sorry for that. Asciminib on platelet activation, thrombus formation, and thromboinflammatory responses in chronic myeloid leukemia. Mushab.
It's a BCR-ABL tyrosine kinase inhibitor, SMNE, to see its induced platelet activation, increase thrombus formation, and promote thromboinflammatory state over time. Most of tyrosine kinase inhibitor are ATP pocket inhibitor, and many patients induce relapse intolerance and resistance against ATP pocket inhibitor. That's why asciminib is invented. Asciminib is an ATP pocket inhibitor, but allosterolic myelostriol inhibitor, and it's related to thrombocytopenia. That's why it's important to investigate its ability to induce platelet activation, increase thrombus formation, and promote thromboinflammatory state over time. So in this presentation, I would like to summarize my project finding. As you see in this slide. So asciminib is decreased platelet activation by decreased alpha granule release and show no effect on dense granule exocytosis. Asciminib also decreased platelet adhesion by decrease the level of surface expression glycoprotein. Asciminib also decreased platelet aggregation by decrease the level of integrin alpha 2 beta 3, which is important in platelets interaction. So what I mean by that asciminib is decreased platelets activity, which related to primary hemostasis. Asciminib also decreased in vitro thrombus formation in dose-dependent manner, which is an advantage for asciminib over other tyrosine kinase inhibitor like ponatinib and nolatinib, as both of them increase in vitro thrombus formation. Asciminib also show a possible thromboinflammatory effect, as it is decreased the level of important cytokines interleukin-6 and platelets and endothelial biomarkers such as B-selectin, E-selectin, and beta-thromboglobulin. So asciminib not just inhibit thrombosis, but also show anti-inflammatory effect. But asciminib increase thrombus generation, which is affect secondary hemostasis coagulation process, and that may lead to ischemic stroke and venous thrombosis. So in short, asciminib is inhibit thrombosis. Asciminib is safer than the previous tyrosine kinase inhibitor, as it is inhibit thrombosis and also inhibit inflammation. Thank you for listening, and I hope you enjoy the presentation.
Thank you, Musab, and thank you for being the first to start this contest. Yeah. We'll wait for a little bit. Catherine reminds me to give the time our judges need to finalize their scores. And Zach or Ezra, when you are ready, let me know so that we can continue. Ready? Perfect. Judges are ready. And that takes us to our next presenter, Anna, who is going to talk about forecasting blood cancer therapy success in older patients.
This is Bill Ludwig. Bill was the very first person to receive a new type of blood cancer therapy called CAR T-cell therapy. Researchers took his immune cells, modified them to become CAR T-cells able to fight cancer, and put them back in his body. Bill has lived more than a decade of cancer-free life after he received his groundbreaking new therapy. This type of therapy is more widely available now, and if it helps, patients can live their healthy, happy lives, like build it. But here's the catch. CAR T-cell therapy doesn't work for everyone, and it is one of the most expensive therapies currently available on the market. And it's risky. It can cause cytokine storm, and some patients have died. And for older patients like Bill, it's even harder to weigh up all risks and benefits, as aging limits immune response, which we think may contribute to the CAR T-cell therapy failure. So the question is, who CAR T-cell therapy will work for? That's what my research is about. To answer this question, I looked at the proteins on immune cells of older and younger individuals searching for a biomarker. It acts something like a crystal ball. In our case, biomarker would be able to tell whether a certain person's immune cells are able to make high numbers of CAR T cells. And I found one biomarker. It's named protein 41BB. I showed that people with lower amount of 41BB on their immune cells are able to make more CAR T cells. Make more CAR T cells. And more CAR T cells. So there is a long way to go, but we've made a great start. We still have to check whether CAR T cells are able to kill cancer cells, and if 41BB is able to work with other types of cancers. But we've made a great start. So the hope is that one day, with just a simple blood test, we will be able to tell whether CAR T cell therapy is going to work in any given patient. Bill said, I was looking for a day, a week, maybe a month, but they told me that I have no cancer. So my job is to make sure every person, especially older ones, get their fair shot at winning this lottery.
Thank you, Anna, for this great presentation. And this takes us to Hassan, visualizing retinal microglia as a window to brain health.
So you may hear the quote of Saint Jerome that eyes without speaking reveals the secret of the heart. But today I will present how eyes can reveal the secret of the brain health. So we all know that human, animal, pets, all are the sufferer of the brain diseases, but identification of those brain diseases they're invasive, they're painful, they're costly, and they require a trained person to run through the whole protocol. And even after all this effort, the accuracy is not 100%. So scientists have been looking through some alternatives, and retina that remains back of our eye is also part of our central nervous system, have been utilizing, and it has one more advantage. In the retina, we have the microglia, that's the immune cell that combat against the diseases, and it's the same immune cells that have in the brain. And in normal conditions, that microglia have lots of processes, but in disease conditions, that processes reduced and they come to as a bushy and spherical shapes. Also at that moment, they can engulf the disease material and keep inside of their body. And that has the natural glow, and it's known as the autofluorescence. And that could be different from the healthy one. So by this, healthy and diseased one could be differentiated. And in my project, I have been utilizing rat model, where I use both the healthy and diseased one. And I have found that both in the retina and in the brain, the microglial shape changes to more spheroid. They not remains as longer as the more processes. And what about the autofluorescence that remains inside of the soma of their body? So it's also reduced both in the retina and brain, and they maintain a good correlationships for the healthy one, both in the retina and brain, but that correlation disrupts in conditions of the disease occur. And by this, we can differentiate between the healthy and diseased brain very easily. And in future, that findings will help to create a future technology that will detect brain disease within a flash of second. And it will also reduce the diagnosis cost, also time requirement for diagnosis, and overall it will have a positive impact on the economy, society, and medical diagnosis techniques. Thank you, everyone.
Thank you, Hassan. Our judges, give them more time. Perfect. So we have here until now AC-minute platelet activation, blood cancer therapy for older patients, brain health based on microglia. And this takes us to our fourth presenter, Anil Kumar Maheswari, 4D knee imaging technologies for musculoskeletal motion analysis. Anil, are they here? Next to Amber. So, perfect. Which means Baha. How gastrointestinal macrophages respond to life states, diet, and disease.
Have you ever heard a quote saying a healthy mind in a healthy body? Yes, maybe. That's all right. I want you to imagine that your gut is a crowded city full of life, traffic, and constant communication. Now, I want you to imagine that your gut, which is the city, has its own security, and we will call it macrophages. These immune cells keep the peace, removing threats, and run everything smoothly. But what happened to these cells as we age, change our diet, or develop a disease? That's the focus of my research. Gut diseases are very common, yet macrophages play an important role in these conditions. However, it's still not very clear how these macrophages shifting their characteristic to become a pathogenic. This is why my research on how these macrophages respond under these conditions. In my work, I use advanced genomic technology, imaging, and animal model to characterize these macrophages under these conditions. By the end of the project, I aim to identify early sign of poor gut health. Also, we are aiming to go beyond the lab by helping doctors to treat gut inflammation more effectively, giving advice for nutrition, as well as helping life equality for people who had gut chronic diseases. So why you don't see these cells? They are working very hard every day, every moment to protect you. Therefore, my project is helping us to understand how to support them at the right time in the right diet. Thank you.
Thank you. Thank you so much. And this takes us to judges. Are we ready? Can we progress? Yes. Thumbs up. This takes us to the next presenter, Mary Slyo, Invisible Injuries, Visible Glow.
Imagine if, after a head injury, your doctor can simply look at your brain and tell you exactly what's wrong. No expensive scans, no painful procedures, and no confusing mental tests. Just a natural signal pointing them in the right direction. That's the future that I'm working towards. My research looks at something very fascinating happening within our brains. tiny, natural, glowing blobs that appear when the brain is injured. This isn't science fiction or special effects. It's just a natural signal that happens in our brain in response to trauma. Right now, diagnosing traumatic brain injury, like a concussion from a car accident or a sports fall, can be a slow and confusing process. Doctors will often rely on symptoms such as dizziness or memory loss, which can vary from person to person. In some cases, the true extent of the damage can still be hidden. My research focuses on autofluorescence and uses preclinical models of injury. We shine a red laser at these brains, and just by observing the patterns of this glow, I can tell not only if the brain is injured, but where that injury has occurred and how severe it might be. Think of it like walking into a dark room and turning on a laser. As a neuroscientist looking at brain injury, I can tell exactly what's damaged and what's not, just by what lights up like a Christmas tree. Here's why this matters. Faster, more accurate diagnosis could help save lives and help people get the right treatment sooner. It could mean less unnecessary scans and a clearer path to recovery for millions of people every year. Now, we're obviously still in the early stages, but the idea that our brains could already be giving us a natural signal as to what's going on, natural glow in our brain just waiting to be read, fills me with hope. Because sometimes to solve the biggest medical mysteries, we just have to learn to listen to our own bodies. Or in this case, learn how to see the invisible injuries using the visible glow. Thank you.
Thank you, Mary. That was great. Just summarizing what we've heard a little bit until now, we are around the middle of our presenters. We've heard about asciminide platelet activation, blood cancer therapy, microglia-based brain health insights, gastrointestinal macrophages, and how they respond to life states, invisible injuries, visible glow. And our judges, are you ready? Perfect. And that takes us to our next candidate, Samson Ma. Cutting edge science, literally tiny spikes that take down viruses.
Oh, sorry. I think I might have contaminated this microphone. But have you ever wondered what sort of tiny pathogens you might pick up on your hands when you're touching these microphones that's been contaminated, or when you're holding on to handrails in the public transports? You see, studies had shown that viruses can survive on common surfaces up to several hours or even several days. And these viruses can transfer into our body if we unknowingly rub our eyes, nose, or lips. And what's even worse, during COVID-19 pandemic, we were reminded how virus can easily spread. We were frantically spraying all the surfaces we can see with all the disinfectants that could potentially leak to the environment. However, what if I tell you that surface itself can fight back these pathogens? My name is Samson, and my PhD research is about exploring a new way to combat this virus on surfaces. Not using any chemicals, but cutting edge material science, pun intended. So this, more specifically, I'm using nanospikes that are 50,000 times smaller than a single strand of hair. You know, these structures were actually inspired by nature itself. Did you know that insect wings like dragonfly wings, if you zoom in onto their wings, they have similar structures that can literally tear bacteria apart? And I've taken this idea a little bit further, and I engineered this into common materials like metals, plastics. And of course, I tested their antiviral capabilities by recovering these viruses onto cell-based assays. And using powerful electron microscopes, I could visualize these viruses. Specifically, I use influenza virus, and they were caught on top of these nanospikes. Some are pierced, some are torn, but many of them completely crushed and inactivated. And the best performing surfaces reduce infectious viruses by 90% just about in an hour. So what does this mean for the real world? Imagine self-sanitizing phone screens. You go to the toilet seats that are a bit cleaner, and of course, hospital trays and surgery items that will remain sterile, and most importantly, reduce transmission in the community. My research is still in the lab, but the potential is real. In a world where the pandemic could be just lying around the corner, I believe surfaces shouldn't just stay there and spread diseases. It should fight back. Thank you, and this is my research.
Thank you, Samson. Thank you, Samson. Nanospikes around me. Contamination? No, it's not that. I should say that it's really, as a more senior academic, it's really sometimes intimidating to see the talent that grows through our school. I used to think that we are just a powerhouse of research, but it seems that we are a powerhouse of courage and presentation skills. And thank you for reminding that. You are all of you. You are raising the standard for all of us too. And this takes us to judges. Are we ready? Perfect. This takes us to our next presenter, Munaf Hossain, Building a Healthy Brain City, the role of glial-derived quinoerins in perinatal brain development. Thank you. Yes.
Imagine your brain as a bustling city. It fills with network of roads and buildings. In that city, there are two main type of workers. One is neuron, another one is glial cells. Neuron are communication expert. They send and receive messages and ensure everything runs smoothly. On the other hand, glial cells are brain support crew. They provide essential support and ensure everything in place and functioning properly. So let's think about how a safe and well-constructed building comes together. It takes a team of skilled workers, and together they build a structure that is secure and strong. And in our brain city, there is a special construction process called chironine pathway. And this process involves several skilled workers, and each skilled workers derivated by our brain support crew. In my research, I am using mouse model to investigate that special construction process. And in my research, I found that one of the key skill workers is chironic acid. And this chironic acid acts as a safety inspector on a construction site. And it's ensure everything is balanced and maintain burden from damage. On the other hand, another key skill workers in our brain city is quinolic acid. And this quinolic acid supports to build essential structure, but can be harmful if not properly regulated. Much like as a construction project going wrong if the skill workers are not coordinated properly. In my research, I am focusing that skill workers, how they work together to maintain a strong brain city. And I already developed a method for monitoring their function at the early age of brain development, how they maintain their function to keep the brain city strong and secure. By investigating this process, I hope to find out a new ways to protect your baby's brain, my baby's brain, and ensure every child has a best start in their life with a brain city that is strong, healthy, and ready for lifetime learning and growth.
Thank you, Munaf. And that reminds me that we literally had babies in the room before, less than a year old. And yeah, perfect. Judges, are we ready? Perfect. And this takes us to our next presenter, who is going to be a recorded presenter. Yeah, I would keep your phone with nanospikes or without. So she's not able to attend today because of her parental duties. She has 2 kids, and therefore we recorded the. Yeah, we'll do that at the end. Catherine reminds me. You have how many? 4. You're right. I can't stand 1 and you have 4 and you are having a PhD too. It's like carrots. And that takes us from a research which addresses the needs of babies and perinatal health to a mother. Recently, come on. Carly Rose McLaren, The Devil's Dose, investigating radiation therapy as a treatment option for Tasmanian devil facial tumor disease. Please come.
Okay, hi. We all know cancer is horrible. It can affect anyone at any time, anywhere. Now, imagine a cancer that you could catch from a friend or a family member. Imagine that had no treatment options available. That's exactly the disease that's facing the Tasmanian devil. The Tasmanian devil is an endangered marsupial native to Tasmania in Australia. And for the last 30 years, it has been battling a disease called devil facial tumor disease, as you can see here. It's a rare transmissible cancer and is spread through biting. And that biting is part of their everyday life for eating, for breeding, and just for basic survival. This disease pops up tumors on the face and around the mouth and then metastasizes internally, which ultimately becomes fatal. Currently, there is no treatment options available for this disease, and the only research ongoing is focusing on immunotherapy as a preemptive defense against it. And now this disease, because it has no treatment options, has progressed across Tasmania since 1996 when the disease appeared, and has continued to decimate the population until a second strain appeared in 2014. So since I was a child, I've really loved the Tasmanian devil. I'm sure, like most Australians, they're cute and cuddly until they're not. And I'm a radiation therapist, and I see the potential to help. Radiation therapy has not been investigated in the literature for this type of cancer before. And I wanted to know why. So I started to look into the details, into the databases, and it just simply wasn't explored. Now, why wasn't explored? Why wasn't it explored? When this disease appeared 30 years ago, it was very difficult to treat anything. The treatment margins were large. The way that the animals would have to come in would be for many weeks at a time. And that's just not good for a wild animal. So now with modern advancements in radiation therapy, we can have smaller margins, larger doses delivered in a single dose. And now I want to see how this will work. My research is investigating a novel way to treat this disease and checking the responses to different types of radiation and different doses. And I'm going to be coupling this with existing small animal models to see how the population can be improved and how we can save this small Australian icon. With the right science, we can give them a chance to survive, and I hope my research can do that for them. Thank you.
Wow. Very, very interesting line of research and projects we have heard until now. And that takes us to Xinue, the adaptive immune response to influenza, a virus infection.
Okay. Hi, everyone. Influenza infection is a common respiration virus infection that can cause seasonal epidemics globally. So, do you know how our immune system responds to this influenza virus infection? So, our innate immune system function as the first defense wall. So following the detection of the influenza A virus, the innate immune response can use the dendritic cells to produce cytokines and interferons that will facilitate the dendritic cells maturation into the antigen-presenting cells as part of the adaptive immune response recruitment process, which further initiate the immune B cell and T cell responses. So activated antigen bearing dendritic cells can further trigger the differentiation of the naive CD4 T cells into differentiation into its subsets Th1, Th2, and follicular helper T cells. And the follicular helper T cells can activate B cells, which later forms the long-lived memory B cells and also help in generating the antibodies. T helper 1 and T helper 2 cells can regulate the differentiation of the CD8 T cells and indirectly help the maintenance of CD8 T cell responses. The CD8 T cell response T cells can rapidly expansion, differentiation, and migration in infected cells, which further forms the immunological memory T cells, and also in the infected cells, the cytotoxic T lymphocytes responses can further initiate the secreting of cytotoxic granules and other cytokines, which initiate the apoptosis. So our study is among the first to explore the adaptive immune responses in driving the cardiovascular complications in pregnant women by integrating this influenza infection and the immunological changes in pregnancy. So thank you, everyone.
Thank you, Zinue. Judges, are we ready? Do you need some extra time? Perfect. And this takes us to Vinodia, from clamps to clarity, the mission to keep the antibodies stable.
Most of you have cooked rice in a rice cooker where the rice grains are well separated and fluffy. There may be days where you forget to put that in the fridge, but you just left it on the kitchen bench. So on a hot summer day, where these rice grains will stick together to form the clumps. Well, now imagine this rice cooker is a bottle of medicine with a syringe. So just like the rice grains are the antibodies which are inside that bottle. Well, the antibodies are life-saving medicine given for cancer. So once these medicines are made, they will be released from the factories, and before it gets into your hand, it goes on a long journey from one warehouse to another. So under the heat, just like the rice grains, these antibodies will stick together to form the clumps. So actually, you won't see the clumps in this bottle. So once these clumps are formed, that is the real danger. Then these life-saving medicines would become life-threatening ones, causing real danger to the patients. And the big issue is the pharmaceutical industries still struggle to find the answers to this problem. That's because the clumping or sticking of these antibodies is not something that's easy to understand, and so they are still struggling to understand this issue. So to prevent this clumping or sticking of the antibodies, they added special ingredients. So these special ingredients is actually acting like the bodyguard. They protect these antibodies and prevent this clumping. So here's where my research comes in. So actually, and the pharmaceutical industries also spend billions of dollars to figure out what's the best recipe to tackle this issue. So here's my research comes on. I'm dealing with identifying, I'm also trying different ingredients, but actually I'm developing a model so you can forecast what would be the best way to prevent this clumping. So actually, a little medicine inside this bottle is very important because it's someone's life and someone's hope. Thank you.
Thank you, Vinodia. Um, are we ready? Seems the judges are very fast or very unanimous in how they judge things. And now we have, yeah, the last. Yeah. How do we do that? Okay. So we just need to wait for the recorded presentation to occur. I'm just contemplating what we have heard until now from the beginning of this present, competition till the end. Asciminid platelets, microglia-based brain health, autofluorescence, which makes problems visible, nanospikes, and surfaces which need to be made flat, blood cancer, Zinue. We've heard about a series of topics addressing health of babies, developing individuals, older individuals, animals like the Tasmanian devil, and now we are going to hear about social media and how, interestingly, in a recorded presentation.
My name is Vania. I'm 13 years old. My phone has been taken away from me, so I no more use TikTok, Snapchat, and Instagram. I'm sad. I'm gloomy. People think they can discipline me, but no. I'm feeling low. I'm feeling alone. I want my phone back. Please return me my phone. I'm in the dark. I don't have a way forward. This is where my research comes in. My name is SHBS from School of Health and Biomedical Sciences. I work with the largest data from ABCD study, that is massive project tracking over 12,000 kids across United States as they are growing up. Using the cutting-edge modalities of different tensor imaging and structural MRI, that is fractionalized 2P and T2 weighted images, I study the white matter of interior cingulate cortex, manage emotions, and resist temptations like, say, checking your phone for the 100th time. Here, the neural highways are structured. In simple terms, the stronger the connections, the better the brain can regulate attention and behavior. You must be thinking, why does it matter? Let's say you are going to your office and you are spilling the fuel, water, and oil of your car or over the road. You are not only doing this dangerous for you, but for the people around you. This is called as the axonal degradation due to social media addiction that is happening at the moment. You must also be thinking that why does it matter? It does matter because adolescent is a critical window. The brain is still wiring itself, and that wiring is shaped by endless digital feedback, loops, likes, shares, endless scrolling. It might change how teens handle emotions, attention, and even risk silently. This isn't about blaming the phones. It's about understanding the biology of this behavior. If we can identify the neural signatures of social media overuse early, we may be able to intervene through education, mental health support, and even how we design safer digital platforms. My goal is simple: to decode how social media affects the developing brain so we can protect it, guide it, and give the next generation a chance to thrive both online and offline. And for that, we need your support. Thank you.
Now judges will have some time to deliberate, conclude the winner, and the audience will have time to vote. It's going to be a very hard process, voting the best out of these presenters. And at this point, I should probably say thank you to all of you who presented today, each and every one of you. Thank you to Samson, thank you to Munaf, thank you to Baha, thank you to Vinodia, Anna, Musab, Hassan, Mary, Zinue, Kanyual, who is not here, Kali at the back with her kids. You've all been extremely inspiring, brave, and gifted, talented to showcase your research, to pitch your research in front of an academic audience. It's a great challenge. We all know how difficult that is. It's a great honor and privilege to be chairing this competition, to be presenting this competition alongside each and every one of you. Thank you for what you bring to our school, the inspiration and the growth you bring with you. It's great. And I personally admire, as I said, each and every one of you for what you just did, which is extremely challenging and difficult at your level. While many of you may not have presented your native tongue, many of you may have kids and, had to juggle with concurrent obligations, not only for your PhD, but to be present here. And it's greatly appreciated on behalf of the school, greatly appreciated for what you bring with you. Thank you to your supervisors for continuously supporting this process. Some of them are in this room: Tien, Sarah, Mary, Bobby, Kylie. Thank you. Thank you to all of you. Thank you to our judges who invested their time, and they are all very thankful to be here with us without any extra benefit. Thank you to Oren, thank you to Ezra, and thank you to Jack. And yeah, I think, have I covered the time that you need to conclude? And the most important thing now, thank you to Catherine, to Annie, to Cody, and to Nicole, who have been organizing the 3 minutes competition across the STEM college. And we are the 4th competition within a week. And it might be stressful for the presenters, but believe it or not, it's very much more stressful for the organizers to bring us all together to facilitate the process the right way. And I think, yeah, have we all finished voting? That's quick. And that gives us time for our keynote, Michaela, who won the school's 3 minutes competition, then the university's 3 minutes competition last year, to give us a special address.
Thank you. How do I? Oh, yeah, perfect. Okay. Oh, it's gone a bit small. That's okay. Thank you so much for the warm welcome, and well done to everybody who presented their 3MT. You guys definitely raising the bar this year. You all did a fantastic job, and you all have such wonderful, interesting research projects. So I would like to welcome you to what I like to call the Researcher's Guide to Research Elevator Pitches, presented to you by a researcher for researchers. Look, this is a disclaimer. It can apply to any kind of multi-level transport, stairs, elevators, escalators, travelators, if you're so inclined. It doesn't just have to be an elevator. Now, I was asked to come and give the keynote so that the judges would have time to write up their report. So I won't keep you too long. I know we're all excited to hear the winner. And I said to them, you know, what do you want me to talk about? Talk about anything. And they said, free reign. Right. So any of you who have seen me talk know that I like to try and inspire and motivate and keep things positive and very lighthearted. And so I thought, you know, I don't take myself too seriously. That is my preference. And what better thing to talk to you about today than, you know, my academic journey coming through my bachelor and my PhD, having just finished my PhD about a week ago. Also, thank you. Also, just to tinse you a little bit about my research, of course, but everything I love so much about the 3&T competition. So what I'm hoping that you will be able to take away from this today is that presenting can and very much should be very fun. And I do hope that some of my tips and tricks you will be able to take and adopt into making your own presentations in the future. Okay, cool. So when I started my journey here at RMIT, I actually enrolled in a Bachelor of Applied Science and I majored in psychology. I chose this major because I was good at psychology back in high school. I didn't really have any other passions. I just really liked knowing how things work in the brain. And I thought, yeah, why not? But as I moved through my bachelor and I sort of got exposed to more of the courses, I thought to myself, you know, I think I really like neuroscience. So as it turned out, I didn't really care all that much about who the father of the psychology was, although I do know that about but I cared more about exactly how the brain works. I mean, what's happening inside of the brain that is changing the brain to change our behavior. You know, I wanted to explain that behavior that I was learning about in psychology, but at the biopsychology level. So when I started my honours, there came a time to do a research project and I reached out to the Health and Biomedical Science School. I reached out to my supervisor, Mary, she's in the audience, and I said to her, please, 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 Mary, do you have a project that involves the brain? I want to do research. And that yes was a defining moment for me. So I won't lie, I hadn't done any kind of biology research or any kind of biology courses since high school, but I did know that the mitochondria is the powerhouse of the cell, and I just figured I could build on that and wing it. So I did my project, I wrote a thesis, and I enjoyed it so much as I could, given that COVID kind of limited the time that we could spend in the lab, but I had such a great time. And then I was given another choice. I could either continue with psychology and try and get my master's and become a psychologist and listen to people's problems day in and day out, or I could do a PhD in biomedicine and still listen to people's problems day in and day out, but I would have a PhD. So, you know, Mary's very convincing, and she obviously had a lot of faith in me. So that brings me to today with a PhD, and I must say it was the best decision of my life. Whoops, sorry. There we go. So what did I do in my PhD? My research was mainly focused on the question, how does the brain get its folds? Now, there are lots of different ideas in this area, but ultimately my research focused on the fact that there are genes in the brain that are more or they get less expressed in some areas, and this correlates very, very well to where maybe a fold will grow or where it won't. And so my research was able to then find novel genes, which we thought might be responsible for how the brain gets its shape. So I took that data and I made something that I like to call my Franken gene, and it's kind of a mashup between a human gene and a ferret gene. You smush them together. And this particular gene that I chose that I really liked was supposed to be a cortical folding gene in humans. So I gave it to ferrets. Why? Because I wanted to know how it worked and what would happen when it doesn't work. Now, why do my research at all? Well, because changes in the way that the brain gets its folds can be associated with clinical disorders. It can be associated with getting epilepsies, intellectual disability, schizophrenia, even autism. So by understanding which genes and how these genes contribute to the formation of the brain means that we can better understand when things don't go quite as right as they should in development. And this means that we can create better and earlier interventions for patients and the community. Now, like everybody in this room, I wholeheartedly believe that my research is the most important thing in the world. But I could spend hours talking about all of these hypotheses, all of the cells, the biological pathways. We won't talk about the heartache of the RNA sequencing experiment that I experienced at the start of my PhD. But a key part of being a good scientist is getting others to think the same way about your research and your very, very, very niche area as you do, right? It's about making them care, okay? So the question for me that I'm interested in is, how does the brain get its folds? But I guess what you're all thinking is, why should I care? So why does it matter if the brain gets its folds, and why should you care? Well, what we do day in and day out is we answer questions and we solve problems with our research, right? But in order to get someone to care about the research the way that you do, most times you have to convince them that they have a problem. Second of all, you have to convince them that that problem can be solved. And finally, you have to tell them that you're going to be the one to solve it. Okay, we have to convince them that there is a population that this problem affects and that it is somehow important. This might be socially, it might be financially, right? And lastly, we have to explain that our research, or how our research, is going to have a long-lasting positive impact on the community. So at the end of the day, research is a sale. You're all part of that sale. You sell yourself as a means for achieving the goals of that research. You sell your idea, a promise, an outcome. And, part of our job is convincing someone that the research that you're going to do is about as good as this poker hand. And look, I'm not good at poker, but I would say 5.5 aces is a pretty good hand. Thank you, ChatGPT. So all of this here on this slide, this is essentially your elevator pitch, right? It is the cleanest, most concise, high impact version of your work, and you should be able to deliver it to any and all audiences. This is really the beauty of the 3MT and everything that it represents, and everything I like so much about the 3MT. Now, to be frank, I was a little bit reluctant about going in 3MT to begin with. And at the start of my PhD, in fact, I was pretty reluctant to do any sort of presenting or going to conferences or getting up in front of people and being worried that I'd be seen as an imposter. But, as my supervisor, Mary, she'd say to me, this is part of the PhD experience. You get up and you share your research and you meet people and things like that. And, and I was apprehensive. But she's got this thing going where she says, I won't be mad, just disappointed. And I did the 3MT. Now, I had an absolutely fantastic time competing in 3MT, and actually I was pretty shocked when I won. I remember it very well. There's a video, there's Ross, he gets up and he's announcing the winner, and he says my name, and I very loudly go, "What?" And I was absolutely overjoyed. And the thing I liked the most about doing this was the fact that I got to create a talk and I got to convey the amount of passion I had for my research and give it to everybody else. Now, if there is anyone in the room today that was thinking about doing 3MT but then decided not to for any reason, I urge you, please reconsider doing it next year. In fact, reconsider doing any opportunity that presents itself to you, whether it be conferences, whether it be posters, competitions, especially if there is a cash prize. All of these opportunities are building up your ability to create an engaging talk. They are bettering your science communication skills, and ultimately they're going to improve your presentation skills and your confidence when you get up in front of a group of people like this. So There we go. So what can we take away from today? As I said, please, please, please get involved as much as you can. Competitions, conferences, networking, right? But always remember, know your audience. That is the most important thing. You have to know who you're presenting to, right? Remember, it's all about making the person that you're talking to, your audience, your, grant body, anything. You need them to care about your research about as much as you do. Remember that academia it's a constant 3MT, right? Know how to create your elevator pitch and have one in your back pocket. You won't know when you'll need it, but I promise you'll need it, okay? Think about job interviews, think about networking. You meet some rich billionaire that wants to give you some money to do your research. You've got to have that ready, okay? Lastly, just remember to have fun. Right? I am so happy. I love presenting. If you can't tell, I love presenting. I love sneaking photos and things and jokes into my presentations. And if I even get one chuckle, I'm so happy, right? So please remember that presenting research, doing research doesn't always have to be serious, and it can, and it very much should be fun. And look, that's all from me today. I really hope you enjoyed the competition. It was fantastic. You guys did such an amazing job, and I can't wait to pass on to Vas now to see who the winner is.
The winner together. Perfect. That makes me less stressed. Okay.
It's not meant to be stressful, Beth.
No, it's never meant to be stressful. So we'll start with the People's Choice Award. Okay. Yeah, perfect. People's Choice Award is Vinodia. Come up here. And are we taking a photo now? Yes, we are taking a photo now. Yes. Let's go next to Vinodia, both of us. The previous winner. And perfect.
If you won't be forward, out of the.
Perfect.
There we go.
That's great. Congratulations, Vinodia. Amazing. You will stay here now we are together. Yes, of course. But before we go to the, just to perpetuate the agony, before we go there, the significance of communicating your research outside a grant writing process becomes more and more important, because since AI appeared, there have been questions in funding bodies about how that could be addressed. And exactly the same way we have, verbal assignments or video recorded assignments in classes, there have been some ideas in significant funding bodies of attaching a video recording where one pitches their grant for category 1 funding. And what you do now upgrades your skill set. Let's go to the second one, which is going to be the runner-up. Runner-up. Runner-up is?
It is Mary Slayer.
We need to take another photo, eh? Perfect. Smile.
I actually was, which is slightly offensive.
Perfect. And Michaela, we have this. Now.
A winner. Me.
Yes.
Winner.
5, 4, 3, 2, 1, and the winner is. Yeah. And this is going to be a tradition established for the next year's competition. The winner is going to be here to deliver the keynote.
Oh, no.
Go deliver the awards. Yeah. I'm always the clumsy one. My speech now. Michaela, you need to train Rapson. Samson.
Yeah, thank you.
Thank you, everyone. What should I say? I think we should say that it was an excellent way to finish our week. It's been extremely inspiring to see you here. I think you embody the journey of research, the journey of communication, the journey within this school through delivering outcomes. I think it's been magnificent to have 10 competitors, 10 presenters in this round. Each and every one of you did a major step forward. And it's also been very, very significant to have this gathering as an HDR community with some of our supervisors, the HDR team, some of the candidates, even if we are a portion of the bigger community of HDRs within SHBS and supervisors, it brings us closer, it brings us together, and it helps us understand what each and every one of us does in the research. Michaela, your final word. The final word goes this. And the same we'll hear next year with Samson.
Okay, I could do what you did and do a mic drop before. Thank you everyone for coming, and hopefully next year we have as good a turnout as we did this year. Congratulations to all of you that attended and presented. You did a fantastic job, as we said, and as I said, please, if you thought about doing it, come and do it next year. All right? Done.